and then moved to University of Glasgow to undertake an MSc and a PhD in biomedical engineering. And he then joined the School of Chemistry, where he now works on plasmonic sensors and applications, and is currently an EPSRC UKRI Innovation Fellow. And Affar is going to be talking to us today about new technologies for measuring interactions. So Affar, thank you very much.
Great, thank you very much for the introduction, and thank you very much for the invitation. Really interesting. I do admit, coming from an engineering background, fell into biomedical, then did a bit of cell culturing. So somebody mentioned MDCK cells, that rings a bell. And then ended up doing quite a bit on plasmonics, and I've always had a foot in the nanofabrication and technology. So it's really a pleasure to be able to discuss all this, especially given I'm now moving into sort of application phase of the stuff I've worked over for almost 10 years now. And I'll just sort of give a highlight of where I am now, which is the Chemical Photonics group in the School of Chemistry. And we develop basically metamaterials. These materials are basically something when you start creating periodic structures that are on the scale of the electromagnetic wavelength. So in the case of light, you're looking at on the nanoscales. When you start creating that, you can give bulk properties that don't exist in the bulk. So you can create specific points of wavelength where the material will absorb or scatter. So you can make gold look in different colors, for instance. And that's all based on how you structure these things. And then you can apply them for many purposes. And that's a lot of what we look at, which is in the case of, we mess around with some unique materials on top of our structures and see how they interact and all. But that's all the novel bit. And the majority of what I've taken up is the biosensing now. And I'm trying to get all of that applied. And the last 3-4 years actually have all been about trying to get this stuff going. And because we've, these metamaterials that we make, we work on with using metals and we work in the visible wavelengths, they come in the category of something called plasmonics. So I think a lot of, maybe quite a few of you have used an SPR system from Biocore or Horiba, etc. And these work on a similar principle. You essentially have a plasma of electrons, and when you excite them in specific conditions, you can create oscillations or resonances. So they're very much frequency dependent. And in the case of SPR, it's over the surface. If you start taking small nanoparticles, you can have them, they work a bit like antennas. So they have a certain resonant frequency at which they'll scatter light, absorb light, and do different things. But the important thing here for biosensing is that these resonators, their resonance positions in the over the spectrum is highly dependent on the surrounding environment. And that's the principle of how a lot of these biosensors work, is that when you change the refractive index, that resonance position shifts and they then detect that something is stuck on the surface or not. Now, they need to be really surface sensitive, otherwise they start detecting bulk stuff. So it can be quite tricky sometimes to work with things like ceriminol. And we started working with creating chiral nanostructures to try and mitigate that problem. Essentially, when you start creating chiral nanostructures, just like sugars or proteins, you have chirality involved in there, and then they start producing optical rotation. So if you think about polarized light, when you hit that on these surfaces, at specific wavelength, it's going to rotate those polarization and send them back. So in the case of a left-hand structure, you get one kind of spectrum or optical rotation dispersion. In the case of the right-hand structure, you get a slightly different one. But when doing biosensing, we also realized that one of these samples, initially they used to be made through nanofabrication, would just cost about £1,000 each. And that just doesn't work if you want to do these loads of experiments. So we managed to get this into a sort of Blu-ray process, which was optimized to create these nanostructures. And now we can create them on plastic slides, so just polycarbonate. And you can create a lot of them. You can make about 1,000 of them in a day, essentially. We literally make only 200 and we use them over a month. So it's mass manufacturable now. As I'll show, we can perform biosensing using these quite well. Now, one of the big reasons of getting these chiral nanostructures was that the chiral fields, they generate these sort of chiral fields, which are slightly different to what SPR, for instance, does. And the principle here was that that would potentially tell something about the chiral molecule that you stick on the surface. And of course, when the molecule changes its structure, that overall chirality might change, and we might see that and detect that structural change. So we get rid of the whole refractive index detection kind of concept, and we move into trying to understand the molecule that's stuck on the surface and how does its structure change. So we did a lot of this summer, and I think it's kind of when I joined 2015 or so, sorry, a year after I joined. We looked at enzymes binding to small molecules, which are usually quite difficult to detect because of the size of the molecule itself. Doesn't really contribute too much to the refractive index changes. Back then, the technique was very much on the basics. So we used to get these graphs of the ORDs, look how they shift when these sort of interactions were done. And then you could sort of get some of the more simpler data to understand. So if you have a ligand, you have very small asymmetry. The asymmetry is essentially just like any resonance. We look at the shift in one hand of the structure and subtract it by the shift of the other hand of the other-handed structure. And that gives us some what we call an asymmetry factor. And if you have ligands, you get very small because these are tiny. So we can't necessarily go down to very small molecules to apply this process. But when you start looking at the proteins themselves, you get very large. And when they change their structure, you get a change in that. Now, these were all in a very controlled environment. You know, the repeatability, reliability of this is very much dependent on how the protein orients itself. So very difficult thing to do. Either way, we also applied it to detecting orientation of proteins, something very tricky, I guess, to do from what the chemists tell me. But yeah, it kind of, we had to apply a lot of maths here and a lot of theory to achieve this kind of data where we look at how the graph actually changes. So the asymmetry factor here is tiny when you measure it between these different orientations. We took an aphamer, which this company that we work with, Avacta, provided with, this was a cysteine termination. So we expected it to lie in different positions on the gold potentially. And all we were looking at, well, can we differentiate between 3 different positions? Now, these could very well also be to do a bit with the structure and how these proteins interact with the gold when they're oriented in that manner. But we kind of sort of showed that there is potential of detecting it. We then went on to do some plant viruses stuff, and this was just published last year, where putting these plant viruses, we wanted to see, can we detect the overall interactions and stuff? Can we make some sort of assays? And surely we found that, especially with the viruses on, especially if they're ordered in a particular fashion using fab fragments, you can then get some very large differences. Now, all of this by 2018, I was sort of looking at my fellowship and stuff, and my biggest aim in all of that was the fact that we do a lot of immunoassays, okay? And if a lot of it's done through people working hard in the lab over a few hours, getting this whole process done. There are loads of technologies, I agree, that are now looking at how to mitigate these problems, make it simpler, so people don't have to really have expertise. You put these cartridge in systems, even PCR and all. But one of the things that really gets me going with immunoassays, it's still dependent on multiple things. You've got to have secondary antibodies, you've got to have fluorophores, you've got to make sure everything works right, sample sizes increase, multiple steps, and basically you need to have an expert doing all of this, otherwise it's very difficult to necessarily make it run. And so my interest was to mitigate these steps and make something which is very simple so that we can provide diagnostics more on the level of how we now provide people coffee. So you can actually do this in your house, you could do it in a clinic, and that's the aim of the whole project. But to do that, especially if we want to detect a disease, you may have to detect multiple markers. And we had to get into the whole point of that this whole system cannot just be measuring one thing at a time. We need to start measuring multiple things. So back then, the aim was, okay, can we detect 6 markers for sepsis? Can we detect different fungal infections through certain markers as well? And bovine mastitis was something that popped up with a collaborator of mine in the vet school, which was rather interesting to know that this kind of things also happen. So we started looking into how do we do all that? And the solution was potentially imaging. And we started working with a company called Horiba, and Avacta was there from before as well, providing some binders. And we now lay down our arrays, okay? So each of these squares is 300 by 300 micrometers, and this whole thing is 3 millimeters. So really, really small, about, I think, 3 ballpoint tips essentially. And each of these, we've got 9 of them, so 9 sets, and then inside we've got a left hand and a right hand structure. And if you don't want to do chiral measurements, you can do just plain measurements on them and take averages, and you've got essentially 18 data points from a single experiment. And we can, through imaging, we can do all the measure all the polarimetry and everything. And a lot of time was spent developing the instruments because they didn't exist on the market. So we had to sort of custom make these. And this is kind of what they look like now. So you've got one of these, this is an SEM of these structures. Inside each of these arrays, you've got 160,000 nanostructures that look something like this. They're mass manufactured, which isn't always easy given the scales. We're looking at these arm widths being between somewhere about 10 to 30 nanometers. Okay, so getting that kind of quality every time with a process that's making it in 6 seconds isn't trivial. Our sample kind of looks like that. You can see we've laid out almost 6 different sort of, you can have 6 sets of these whole sets of structures laid out. And now what we've done is we've even added in some sort of custom-made microfluidics. So you don't have to have, like in SPR systems, you don't have to have microfluidic channels or flow systems or anything. You can literally just pipette in a port on this side. There's a small 3 millimeter, I think 2 millimeter now, designed port where you can pipette something in and out. And you can, it's basically just uses about 120 microliters. And what we've seen up till now is that we can go into tens of nanomolars. We're still working to better understand the limits of detection. It's highly dependent, obviously, on the affinity and the basis of the binding interaction itself, the chemistry itself. So this was, I think, measured using hemoglobin haptoglobin binding, which is quite weak compared to things like streptavidin, biotin interactions, or somewhere in the middle where your antibodies, for instance, would lie. So essentially, we've taken a bit of an evolution on SPR. So anyone who's used SPR would see this a bit of an evolution on it. And so we started applying it. So we have to do quite a bit to get these binders on the surface. You could put in antibodies, you can put in anything. So in this case, for instance, we spent about a day well, not a day, we literally put this stuff now and leave it for overnight, where you create a surface assembled monolayer. You've got to do rinsing and everything. But this is where we want other people, that they only need to be able to do, people who are doing applications such as yourself, to simply have to do the last 2 steps, that you put in your sample, you look for something to bind to whatever's on the surface, and you get your result. And you can even do kinetics through these systems as well if required. So just some of the basics things that we were sort of doing last year, which is to look at streptavidin biotin binding. And you can see that as you go from your, these are not asymmetry. This is just looking at resonance shift. So we're no longer looking at chirality, but just seeing, can we detect things binding? And you can see that from when you go from buffer, your self-assembled monolayer has a bit of it, but this is the step where you now have streptavidin. The self-assembled monolayer has biotin that is anchored onto the surface. So you can now start detecting that. And we further added biotin in to see, well, can we actually detect asymmetry or structural changes in the streptavidin? The asymmetry was very spread out. There was a lot of variance. But if you look at it, always changes shape. And through literature, what we understand is streptavidin does contract when it starts binding to biotin. It's got 4 sites, so even if it's bound by 1, it's still got 3, and it further contracts that possibly causes this to happen. Okay, so I'll go a little quicker. The fluorophore we also use fluorescence on the surfaces to try and understand of the homogeneous coating. There's a lot of work on the chemistry involved to achieve good, reliable success over the entire area. And we can also use the fluorescence enhancement properties of the plasmonic. So you can see there's protein everywhere, but it's just a monolayer. So you can't necessarily detect it very well. But on the nanostructures, you have about a factor of 10 enhancement of fluorescence. So that is another property that can be used. Okay, so now the last bit is basically something we're now working with Ed here. And that's to do a proof of concept of whether we can detect multiple viruses in such a setup. Can we create an assay that can actually look for not just SARS viruses, which is a lot of what the lateral flow devices are doing, but if somebody comes up with a cough and cold-like symptoms as well, can we just scan them for everything to make sure, okay, if he does have influenza, then you can be confident. If he has none of them, then perhaps you want to potentially do PCR and find out a bit more. And we start off, obviously, we need to prove that we can do multiplexing. So in this image here, you can see that you've just got that 3 millimeter area, and we've put 4 spots. The top 2 are for S protein, and the bottom 2 are functionalized with streptavidin. And then we introduce the antibodies individually for each of them. And you can see that in the case when we add the streptavidin antibody, it's only the streptavidin that jumps up in its resonance. And in the case of the S protein, it stays the same. And now with the S protein antibody added in for the S protein, you can see a change there. Okay, so we can be quite specific in finding out what's binding to what here. And then we've just done these recently, where we've looked at in sort of a buffer with a bit of mucin mixed in it to try and replicate kind of similar simple what kind of sample-like material that we'd have to use later. And you can see that when we add in S protein, so when you add in just a control mucin, so there are a bunch of proteins floating around, they don't interact, it's quite specific. The moment you add in, you then basically see the S protein jump up, and you can sort of get a very big shift here. And the same case for doing it with hemagglutinin. And here in the S protein, we had affimers, but in the hemagglutinin, we're using Fab fragments. So it's very flexible. You can put whatever you want to work as your binder and to detect specific targets in this case. So last, I'll just quickly end with saying, well, some of the ideas are what kind of diagnostic assays that may be useful or people interested. There was talk about BLI and how we can sort of perhaps do something that helps you on those aspects of BLI that it might struggle with. Can we also, with the nanofabrication, develop something where we can trap things in holes, nanoscale sized holes and things that may be of interest, and then also detect them through these kind of similar principles? Or could we even think about creating a bioreactor-like system where you have regions of incubation of viruses and then see how perhaps when they reach a certain point, they start proliferating or sending out other proteins that can be detected in another region of those reactors? A lot of our skills are around nanofabrication, and we've now got systems for confocal Raman spectroscopy and AFM in our own lab. So this is something, if somebody's interested in, we'd certainly like to sort of talk about and collaborate on. And last, I just want to quickly acknowledge the School of Chemistry and my current team of Cameron, Martin, and Stephanie, who've done a great job. And we work with these companies here. Avacta produce affimers. A few of you may have worked with them as well. And these are our funders. Thank you very much.
Thank you very much, Affar. It's really interesting to see that and the steps through to viral applications. I think once again, we're going to have to keep questions quite short just to keep to the schedule, but does anyone have a quick question for Affar now? We can perhaps revisit more of those later on. Would anyone like to ask a question now?
It's all right. I feel the same when they talk a lot about the biology. It's just...
You see, I have lots of questions, but of course we do collaborate, so I've asked quite a few already. Lonica, I see a hand raised from you.
Can I ask a very simple question as a biologist? What costs are you talking about? When you think about diagnostics, how widely can you apply it because of costs? Which is slightly different in the human field than the veterinary field, but can you give an indication?
So I'm thinking about, for instance, comparing it to SPR, where each chip can cost about 100 pounds. Something like this would cost in the tens of pounds. So we're really trying to mitigate the cost in that sense. And then from the fact that we can use really small amounts of proteins in the case of assays, but to be fair, now the fluorescent systems now work with almost femtoliter proteins and things as well. But if you want to do something a bit more simple and hands-on, then you could potentially use this. And again, we use very small amounts of protein. You can see the kind of spot sizes we have. So hopefully what we tend to do is in one experiment, if you do 100 different markers or disease markers or viruses or something like that, then the overall cost drops because you're doing it over just a 3 millimeter area with very small amounts of, well, for 100, we have to expand it a bit more, but those kind of capabilities essentially.
Okay, thank you very much, Affar.